So our first session is on investigation of vascular Ehlers-Danlos syndrome therapeutics and interventions using mouse models. The first talk will be by Emily Juzwiak from Baltimore, from Johns Hopkins. And the title of the talk is Spironolactone Monotherapy Provides Overt Protection in Post-Pubertal Vascular Ehlers-Danlos Syndrome Mice. So Emily, take it away.
All right. Thank you, Dr. Dietz, for that wonderful introduction. I'd like to say thank you for inviting me to speak this morning. These are our relevant disclosures. Vascular Ehlers-Danlos syndrome is a connective tissue disorder caused by a heterozygous mutation in COL3A1. Patients have thin, translucent skin, easy bruising, and a characteristic facial appearance. People with VEDS are susceptible to rupture of the arteries, intestines, and gravid uterus. Typically, there is no aneurysm prior to rupture, making the disease difficult to predict. This disorder has a median survival of 45 years, and the most common cause of death is due to spontaneous arterial rupture. 25% of patients have a major event before the age of 20. And the greatest risk for an early vascular event is seen in males around the time of puberty. It remains unknown how mutations in a structural protein such as collagen cause disease. To address this, our lab created knock-in mouse models of VEDS. Our mouse models recapitulate VEDS phenotypes. Both models involve heterozygous glycine substitutions in the triple helical domain of type 3 collagen. Both mutations have been previously observed in people with VEDS. And both models show spontaneous death due to aortic rupture, as evidenced by hemothorax or hemoperitoneum in all cases. A more mild model with an N-terminal glycine substitution shows a median survival of about 400 days, while the model with a more C-terminal substitution shows a median survival of only 40 days and 100% lethality by 6 months of age. The majority of this talk will focus on our more severe model of VEDS, but concordant findings have been observed in the mild model. To gain insights into the pathogenesis of VEDS in an unbiased manner, we performed RNA sequencing analysis of the proximal descending thoracic aorta and VEDS mice and compared the results to their wild-type littermates. Importantly, this aortic segment was selected because it is by far the most common site for vascular dissection or rupture in our mouse model. Samples were taken at an age where animals are predisposed to vascular events. There was no evidence of either aneurysm or tear at the time of sample collection. This would allow an assessment of molecular events that are potentially a cause rather than a consequence of vascular catastrophe. As demonstrated by this heat map, RNA sequencing identified a gene expression signature that differentiates wild-type and VEDS mice. Genes that are upregulated are shown in red, and genes that are downregulated are shown in blue. Unbiased computer algorithms predicted upstream drivers of this gene expression signature, revealing the likely involvement of signaling through a G protein coupled receptor that specifically utilizes a G-alpha sub-Q subunit and the mitogen activated protein kinase signaling cascade that specifically involves PKC, MEK, and ERK. The relevance of this pathway was validated through the use of selective inhibitors of PKC and ERK activation, which proved protective in VEDS mice. As previously reported, we initiated a trial of hydralazine, an antihypertensive agent that works at least in part through the blockade of IP3-mediated PKC activation. Hydralazine achieved 100% survival in VEDS mice through the first 40 days of life, as shown by the maroon curve. However, at about 45 days of age, mice treated with hydralazine show abrupt predisposition to aortic rupture. ultimately mimicking the performance of their never-treated counterparts. Notably, this abrupt transition coincides with the onset of sexual maturity. While this effect was evident in females, it was greatly exaggerated in males. The male bias and onset of death at puberty led to the hypothesis that androgens may have a role in VEDS pathogenesis. So what are androgens? They are hormones that contribute to male secondary sexual characteristics. They are present in both males and females, but much higher in males. Androgens peak at the time of puberty in both sexes, but again, this is greatly exaggerated in males. The androgen receptor is expressed in many cell types relevant to the arterial wall, including vascular smooth muscle cells, endothelial cells, resident macrophages, and fibroblasts. To test our hypothesis that androgens contribute to the pathogenesis of VEDS, we treated our mouse model with the potent androgen receptor antagonist bicalutamide. When hydralazine was used to get mice to puberty and bicalutamide was added just prior to puberty to block androgens, as shown by the green curve, we saw essentially complete and sustained survival in VEDS mice of both sexes. Anticipating the reluctance of parents to start their teenage sons on a potent androgen receptor antagonist, we explored the potential use of spironolactone, a widely used diuretic that also has substantial androgen blocking properties. Indeed, spironolactone is often used to treat androgen-related conditions like alopecia and acne. The side effects of spironolactone can include mild gynecomastia in males and menstrual irregularity in females. But these side effects are dose-dependent and are fully reversible upon drug cessation. The combination of hydralazine and spironolactone achieved full protection in VEDS mice of both sexes, as shown by the teal curve. As we previously published, a rise in ERK activation attended the onset of puberty and the enhanced risk of aortic rupture in hydralazine-treated male VEDS mice. As expected, this was attenuated by the addition of spironolactone. We then asked if spironolactone monotherapy was sufficient to mimic this effect. We observed that spironolactone monotherapy initiated at weaning had no impact on female VEDS mice and only afforded modest protection in males. We recognized that many people with VEDS first come to medical attention after puberty is complete. This begged the question whether initiation of spironolactone monotherapy after puberty would be sufficient to achieve robust protection. We observed that initiation of spironolactone monotherapy at 60 days of age, when mice are fully mature and fertile, resulted in essentially complete survival of VEDS mice of both sexes. Indeed, the performance of treated VEDS animals in purple was indistinguishable from that observed in the wild-type littermates, shown here in black. In contrast, a cohort of untreated VEDS littermates demonstrated 40% lethality by the 40-day period of observation, as shown in orange. This trial demonstrated that spironolactone monotherapy, when initiated in adulthood, achieves essentially complete protection in both sexes. Finally, our latest work with spironolactone suggests the following conclusions. Spironolactone monotherapy initiated prior to puberty improves survival in male VEDS mice. Spironolactone monotherapy initiated after puberty achieves essentially complete protection in VEDS mice of both sexes. And finally, these data warrant consideration of a clinical trial of spironolactone monotherapy in adults with VEDS. Thank you to my mentor, Dr. Dietz, and our current and past lab members, and we are so thankful to our sponsors who make this work possible. Thank you.
So the next talk is by Caitlin Bowen. Thank you, Emily. That was a wonderful job, by the way. Caitlin Bowen, she is also in my lab at Johns Hopkins. The title of the talk is A Gene Variant and Mechanism for a Potent Protective Modifier of Vascular Ehlers-Danlos Syndrome.
Okay, thanks, Hal. Can everyone see my screen?
Yes.
Okay. Like Hal said, I'll be talking about a project that I've been working on recently, a gene variant and mechanism for a potent protective modifier of vascular Ehlers-Danlos syndrome. So in this study, we really sought to understand the variability in phenotypes, why some patients with VEDS have events early in life and others can go for decades before having an event. And this variability in genetic disease, at least, can be explained in part by allelic heterogeneity, or like for phenotypes that for people that have null mutations compared to those with missense mutations. But you can also see variability in people that have the same mutation or even people who in the same family. So these differences could be explained by differences in environmental factors or by genetic variation that exists in other genes, which we will call modifier genes because they can modify the phenotype. So infrared strains of mice or backgrounds of mice can be used to identify these modifier genes because we can control for both the allele and environmental factors. So we use two strains of mice called 129 and black 6. And these two strains of mice are nearly genetically identical to each other, but not quite. They have a few genetic differences that can really lead to dramatically different phenotypes, even if they have the same allele and environment. So for example, some of these differences lead to differences in coat color. And so the 129 mice are brown and the black 6 mice are black. So with this idea of genetic modifiers in mind, we introduced our EDS mutations onto pure black 6 and 129 backgrounds and looked at the phenotypic effects. So in EDS, both our mild and severe COL3A1 mutations are associated with early death due to aortic rupture on the black 6 background, which is shown in red. And that's what Emily was just discussing. But remarkably, the same mutation on a 129 background, as shown in blue, leads to essentially complete protection from vascular catastrophe and premature death for either the mild or the severe genotype. And the long-term performance of these mice is indistinguishable from that observed for wild-type littermates. So informatively, these differences in survival were not attributable to differences in blood pressure between the two strains or differences in physical mechanical strength of the aortas, although wild-type aortas are a bit stronger than EDS aortas regardless of background. And these protected 129 EDS mice did not have any more collagen or more intact elastin fibrils when we compared them to the susceptible EDS mice on a black 6 background. And when quantified, some of those subtle differences were really exactly the same in the two strains of mice. So this unanticipated dissociation between the structural tissue integrity and the phenotypic outcome was really quite striking. And it led us to hypothesize that potentially some non-productive cellular response to altered matrix, rather than the actual matrix deficiency, is what determines vascular disease risk in EDS. If this was true, it would really bode well for the identification of pharmacologic interventions. So to explore this hypothesis, we performed RNA-seq on the proximal descending thoracic aorta, which is the most common location of aortic rupture in these models. We compared EDS to wild type on both a black 6 and 129 background. And differential expression analysis revealed a high number of significant differences between wild type and EDS on the susceptible black 6 background. But the same analysis on the 129 mice that are protected revealed that there are almost no differences, meaning that they look exactly like their wild type littermates. Informatively, EDS mice that are half black 6 and half 129, also called an F1 generation, which is shown in purple, show improved long-term survival, suggesting the presence of a genetic modifier that could act in an autosomal dominant manner. So we move forward with the hypothesis that the 129 background has a variant in a gene or genes that can rescue EDS vascular rupture risk in an autosomal dominant manner. So to identify the modifier, we used survival time to stratify the mice into cases and controls, or mice that die from aortic rupture before 8 weeks of age, which is very similar to our susceptible black 6 background, and mice that are long-term survivors, similar to the protected 129 EDS mice. So we bred these Black 6 and 129 mice together for several generations to get a mixed background cohort of EDS mice that all had a random assortment of Black 6 and 129 alleles. Informatively, this mixed background population shown on the right stratified really nicely into these case control populations or susceptible and protected phenotype groups. So using these populations, we performed a genome-wide association study looking for regions of the genome that would associate with protection from vascular rupture. So we identified a single protective locus on chromosome 11. The odds ratio at this locus is significantly less than 1, which means that the presence of a 129 allele at this locus protects mice from early death due to aortic rupture. So we then looked for a candidate functional variant within this locus that differed between the two backgrounds. So this point and the center is the significant SNP from the previous graph, and the two flanking SNPs are shown on either side. And there are about 102 genes that exist at this locus. So within this region, we first filtered for genes that are expressed in the aorta. That narrowed it to 56 genes. We then filtered for genes that actually had functional differences between Black 6 and 129. We defined these as splice variants, functional missense variants, or expression differences. That only left us with 9 genes. But of those 9, one stood out as being particularly relevant. This gene is MAP2K6. MAP2K6 has a missense variant that between Black 6 and 129 that's predicted to affect the function of the gene. In asking if this gene was relevant to VEDS, we knocked it out in the protected 129 EDS mice. And we found that the loss of MAP2K6 made them susceptible to death from aortic rupture, indicating first that the function of MAP2K6 is critical for protection from vascular rupture in protected 129 background mice. And second, confirming that this could be a major modifier in mouse models of VEDS. Our next question was, how might MAP2K6 affect EDS aortic rupture risk? The gene MAP2K6 encodes a protein kinase called M2K6, which together with M3K4 activates another kinase called P38. P38 then acts directly on protein phosphatases, PP1 and PP2A, which are the main phosphatases that can dephosphorylate and deactivate PKC and ERK. So we therefore hypothesized that the protected 129 background would have increased M2K6 activity, leading to increased P38 activation, increased protein phosphatase activity, and therefore a reduction in PKC and ERK activation. As we expected, Immunoblot showed that P38 activation was enhanced in protected 129 background mice, as measured by phospho-P38. We also found that protein phosphatase activity, as measured by a dephosphorylation assay, was elevated in protected 129 background mice, shown in blue, compared to the susceptible Black 6 background mice, shown in black. We also found decreased activation of PKC and ERK in the protected 129 EDS mice as compared to the Black 6 EDS mice. To test the hypothesis that the elevated P38 and protein phosphatase activity protects EDS mice from vascular rupture, we examined the role of P38 activity directly. Here we hypothesized that the inhibition of P38 would increase the risk of vascular rupture, and that this increase would occur in a PKC and ERK-dependent manner. Inhibition of this protective axis by treatment with a selective P38 inhibitor significantly accelerated vascular rupture in EDS mice. This occurred in association with an increase in PKC and ERK activation. The risk of rupture was ERK-dependent, as the increased risk was rescued by simultaneous treatment with a MEK antagonist, which is shown in blue. So taken together, these data illustrate that the 129 background prevents vascular rupture by a variant in MAP2K6 that leads to increased P38 activation and protein phosphatase activity, which normalizes PKC and ERK activation. Inhibition of this protective axis in EDS mice increases the risk of rupture in a PKC and ERK-dependent manner. So quite remarkably, this rescue on the 129 background wasn't really dependent on an improvement in collagen deposition or any inherent changes to the inherent structural integrity of the aortic wall. And instead, this phenotypic rescue appears to be dependent on the biochemical and gene expression response of vascular cells. This really bodes well for the development of pharmacologic strategies that could mimic this mechanism of protection. It also adds confidence regarding the potential of therapies that target this PKC and ERK axis, as all of our findings continue to collapse on this pathway. These results both validate and extend our understanding of cellular signaling events in V-EDS. With that, I'd like to thank our sponsors as well as all the people in the lab, and I would be happy to take any questions.
Okay, I'm happy to present here today, and I would like to thank the organizing committee also for giving me the opportunity for this presentation. So in our study, we also use a mouse model of the vascular Ehlers-Danlos syndrome, and back then when we started this project, there were actually two different mouse models available. The one on top was described as truly haploinsufficient. Homozygous mice showed a spontaneous mortality due to blood vessel ruptures. However, heterozygous mice showed only an increased late onset susceptibility to aortic lesions. The second mouse model was originally described with a mixed background and also as true haploinsufficiency model. However, in these mice, homozygosity is embryonic lethal, and roughly 30% of heterozygous mice die due to aortic dissection or rupture, with the highest mortality rate peaking between the weeks 4 to 10 of age. So based on this recapitulation of the key vascular aspects of VEDS, in heterozygous mice, we chose this mouse model for our project. And we backcrossed these mice to Black 6 and further also characterized and recharacterized it, thereby unraveling the true underlying mutation, which is actually an inferent deletion. In the meantime, other mouse models for investigating the vascular Ehlers-Danlos syndrome has been published, such as the transgene model overexpressing a mutated form of collagen 3 alpha 1, and as seen in the two previous talks just recently, the two glycine substitution models of the group of Professor Dietz, also really nicely recapitulating the vascular features of V-EDS. Nevertheless, as I mentioned, our study was entirely done with the second mouse model from top, and all the data I present today is derived from this mouse model. This slide shows the overview of the entire project. However, I will actually not go into the mouse model characterization, but directly focus on our novel and objective readout of the aortic rupture force as a readout possibility for the biomechanical integrity of the aorta. On the left of this slide, you can see the device we are using for our measurements. This is a tissue puller device, and this device has two pins as shown by the image in the center of the slide, and they are just 200 micrometers in diameter, so that you have kind of a scale idea. On these two pins, we mount aortic segments, 3 per mouse, 1 from the ascending and 2 from the descending thoracic aorta, and this gives us an individual level biomechanical readout of the aortic integrity compared, for example, to the cohort outcome of clinical trials limited to morbidity and mortality due to age or other risk factor related arterial events. So after mounting of a segment, the pins are fixed with 2 metal plates and everything is submerged into a buffer. This buffer is tempered to 37 degrees to best possibly mimic physiological conditions. And in the top row, there is a sped up animated GIF of such a measurement. So the upper pin can then be pulled upwards automatically, and thereby the force generated by the segment is recorded. And we take the maximum force as readout for the rupture force. So in a first step, we obviously had to make sure to be able to measure a significant difference between wild type and heterozygous mice. And as I will actually use this graph to compare different treatments, on the y-axis, we display the relative aortic rupture force, not the absolutely measured rupture force in millinewton. On the x-axis, we have the 3 different aortic segments, S1, S2, and S3, and the arrow bars always indicate 95% confidence intervals. So from this picture, one can actually take 2 messages. First off, for both wild type and heterozygous mice with increasing distance from the heart, the aortic rupture force also decreases, as well as the aortic rupture force is significantly reduced in heterozygous mice. when compared to wild-type mice, which is why we concluded that with this readout system, we are able to detect significant differences in the rupture force of the murine aorta. And we can use this to assess the impact of medical therapeutic approaches with the aim of finding a targeted therapy. So regarding target therapies, there are two main ways to go. One of them is novel drug development. However, this is usually associated with high costs, high risk for failure, as well as a long duration from bench to bedside. The other possibility is drug repositioning, which is also known as drug repurposing. So this basically is the concept of identifying novel uses for already approved drugs that are outside of the original medical intention. And this has several advantages, such as usually associated with low costs. There's a low risk for side effects, as there are long-term studies and a high level of knowledge already gained. And for patients, usually it's an eased up accessibility as the drugs are market approved. So for the study design, which is actually for all the results I'm going to show you in this presentation the same. So after birth, we let the mice get to the age of 4 weeks and weaned them and immediately started the treatment. All the treatment were for additional 4 weeks, and at the age of 8 weeks, we euthanized the mice and performed the aortic rupture force measurement. Here you can see the two first drugs we investigated. Both are antihypertensive drugs, as usually regarding medical therapy or V-Eds, the aim is to prevent spikes in or continuously elevated blood pressure. You can all see the original reasoning. So we tested the beta blocker celiprolol due to promising results of a clinical study in V-Eds patient published in 2010, as well as losartan, so an angiotensin II receptor type 1 inhibitor, showing to slow down aneurysm progression and reduce elastic fiber breaks in a mouse model of the Marfan syndrome, so phenotypically similar disease. And also in humans, it has been shown to reduce aneurysm progression in a clinical study published in 2013. This slide shows now the results of 4-week treatment with celiprolol in orange and losartan in purple. And celiprolol significantly increased the relative aortic rupture force after 4-week treatment, thereby explaining the beneficial outcome of the clinical trial, whereas treatment with losartan did not have any impact at all on the relative aortic rupture force, which is why we concluded celiprolol, but not losartan, has a potential added value in the medical therapy of vascular EDS. So this is an image of a retrospective clinical study published in 2019, again underlying this added value of celiprolol in VDS. And obviously the studies about celiprolol raised the clinically highly relevant question whether or not this presumably beneficial effect can be extrapolated to other beta blockers as well. And in this line, we tested the beta blocker bisoprolol, as it is frequently described here, and thus could be a potential alternative to celiprolol. However, bisoprolol shown in light blue did not have any impact on the relative aortic rupture force, and therefore this potential added value of celiprolol cannot a priori be extrapolated to other beta blockers. Finally, when going to the literature, we've seen that pravastatin, so an HMG-CoA reductase inhibitor, reduced aortic dilation in a mouse MFS model. And in addition, statins are also associated with a decreased risk for dissection, rupture, or death in patients with thoracic aortic aneurysms, making it an interesting candidate for further investigation. Pravastatin is shown in dark blue, and similar to celiprolol, 4-week treatment actually increased the relative aortic rupture force in all 3 segments, and thus, aside of celiprolol, also pravastatin may have an added value in the medical therapy of VEDS. And with that, I'm actually already at the end of my presentation. So as for the take-home messages, our null and objective assay allows to assess the biomechanical integrity, so the rupture force, as readout for medical therapies. Despite phenotypic similarity, the success of medical therapies can profoundly differ between aortic diseases, as shown for losartan. In addition, as shown by the ineffectiveness of bisoprolol in our mouse model and setup, beta blockers should not a priori be prescribed interchangeably. And celiprolol and pravastatin, two actually old drugs with long-term safety records, could have an added value in VDS by strengthening the weakened thoracic aorta. And finally, so this point is also a bit of an outlook. It's not known yet if celiprolol or pravastatin can maintain their added value in case of a long-term study or late onset trial. So we have also some preliminary data that actually the timing might be crucial. And it's also not yet known for VEDS if there's a potential hazardous effect in case of an abrupt discontinuation of taking the drug. And with that, I would like to thank all the people involved in the project, collaborators, the funding sources, and you for your attention.
So our next talk is by Rebecca Sorber from Johns Hopkins. And the title is High Dose Vitamin C Demonstrates a Sexually Dimorphic Survival Benefit in a Mouse Model of Severe Vascular Ehlers-Danlos Syndrome. Becky?
Yep, can everybody see?
Yes.
Okay. Thank you, Hal, for that introduction, and thank you to the Society for providing me with an opportunity to present our research. My name is Rebecca Sorber, and I'm a postdoctoral research fellow in Hal's lab at the Johns Hopkins University School of Medicine. These are our disclosures. As you all well know, vascular Ehlers-Danlos syndrome, also known as Ehlers-Danlos syndrome type 4 or VEDS, is caused by autosomal dominant mutations in the COL3A1 gene. This particular gene encodes for type 3 collagen, which is a critical component for the structural integrity of the vasculature as well as the internal organs. VEDS presents with spontaneous arterial and organ rupture at a young age, with adolescent males demonstrating increased risk in both human patients as well as in mouse models. This phenomena is thought to be due to an androgenic surge during male puberty, contributing to increased disease severity, as was discussed in an earlier talk by one of my colleagues, Emily. Vitamin C is a critical cofactor in the hydroxylation of proline and lysine residues during collagen formation. Specifically, hydroxylation of these residues promotes cross-linking of the individual collagen molecules within the collagen fibril, improving the strength of the overall collagen matrix. Saturating doses of vitamin C are frequently suggested as a treatment adjunct in VEDS, theoretically maximizing the quality of the existing collagen, although there is no clinical or translational evidence currently of benefit underlying this particular treatment strategy. Unlike humans, mice and many other animals are able to manufacture endogenous vitamin C precursors via the hepatic enzyme L-galonolactone oxidase, also known as GULO. Vitamin C deficiency in humans results in the disease process known as scurvy, which is defined by bleeding gums, loose teeth, hair loss, and fragile skin, which represents the impaired overall quality of the connective tissues. Similarly, mice who are total genetic knockouts for GULO demonstrate reduced plasma vitamin C concentrations without supplementation and also demonstrate physical manifestations of scurvy. The research underlying dietary vitamin C recommendations in humans demonstrates that serum vitamin C threshold for preventing scurvy is considerably lower than the threshold at which excess vitamin C excretion is first noted in the urine. The threshold for excess excretion also varies widely between individuals. Mice also maintain a serum vitamin C concentration that exceeds the level merely needed to prevent scurvy and have demonstrated an ability to upregulate vitamin C production in response to increased losses. However, little is known about vitamin C metabolism requirements, specifically in VEDS patients or in mouse models, and if increased serum concentrations confer any benefit. With these factors in mind, we decided to investigate the question of how high-dose vitamin C supplementation may affect survival in a mouse model of severe VEDS. In our lab, as we've already talked about, we utilize a mouse model of VEDS with a COL3A1 knock-in mutation maintained on a Black6 background. This is our severe model, which is characterized by 50% mortality at 45 days of life from thoracic aortic rupture without treatment. Oral vitamin C was administered to these mice at various time points via a supplemented chow for a targeted dose of 20 milligrams per kilogram per day, which is the saturating dose for vitamin C. We first administered vitamin C, demonstrated by the green arrow, immediately at weaning, which is defined in these experiments as day of life 21. Vitamin C supplementation in VEDS mice at weaning led to a significant survival benefit in female mice compared with untreated controls, with 100% survival at 60 days versus 50% in untreated littermates. This phenomenon was sexually dimorphic and was not reproduced in male mice. As you can see here, there was no statistically significant difference between vitamin C treated male VEDS mice and untreated littermates, with both demonstrating roughly 60% mortality at 60 days of life. Next, we altered the time of administration of vitamin C in order to investigate whether some critical window of collagen formation was being missed in the male mice. In this experiment, vitamin C supplementation was initiated at the time of conception by administering vitamin C impregnated chow to breeding pairs and then sustaining that beyond weaning. While the survival benefit conferred on female VEDS mice was again recapitulated in this experiment, male VEDS mice again did not demonstrate any significant survival differences compared with untreated littermates. This indicates that vitamin C alone does not confer a survival benefit in male VEDS mice, regardless of the time of initiation. In order to investigate whether or not sustained vitamin C administration was required for the survival benefits noted in female VEDS mice, we initiated vitamin C supplementation at the time of conception and then withdrew it at the time of weaning. The female mice initially showed a survival benefit until approximately 15 days after withdrawal. Then protection was abruptly lost with the rapid onset of death due to vascular rupture, reaching the expected survival rate of untreated VEDS mice at 50 days. This transient upfront protection followed by a loss of protection likely represents a washout effect of this compound. When compared with the sustained vitamin C administration, withdrawal resulted in a significantly decreased survival, which emphasizes the need for maintenance of vitamin C dosing in order to confer ongoing protection in this model. Because of the known effects of androgens on mortality in this model, we wanted to investigate whether co-administration of vitamin C with androgen blockade would demonstrate an additive survival benefit for male VEDS mice. In this experiment, we administered vitamin C along with androgen blockade in the form of spironolactone at weaning. This drug combination did not improve survival in male mice over spironolactone monotherapy. These results suggest there may be sex-biased differences in vitamin C metabolism which are responsible for the observed sexual dimorphism beyond simply an overriding pubertal androgen effect. In order to investigate potential mechanisms underlying the observed survival differences, we performed assays to examine both collagen quantity and collagen quality of vitamin C treated VEDS mice. First, a hydroxyproline assay for total collagen was performed on aortas taken from mice receiving vitamin C supplementation, which is seen here on the left panel. Neither wild-type nor VEDS mice demonstrated an increase in total collagen in response to vitamin C supplementation. Next, in order to examine collagen quality, we performed an ELISA assay specifically for pure adenylene collagen cross-links using serum samples from VEDS mice. This demonstrated an increase in cross-linking with vitamin C supplementation, suggesting an increase in collagen quality rather than content may be responsible for the survival benefits observed in these trials. Interestingly, there did not seem to be any evident clustering by sex in this analysis, as was demonstrated by the widespread of both the solid symbols representing males and the outline symbols which represent females. In attempting to determine a mechanistic explanation for the potential sex-biased differences in vitamin C metabolism observed in our results, we identified prior research that had highlighted sex differences in the expression of gulo and related enzymes, specifically in inbred mouse strains. We utilized RNA-seq to examine baseline differences in gulo expression between the sexes of wild-type mice on a pure black 6 background, which is the background utilized in our severe VEDS model. We noted that the livers of female mice demonstrated significantly lower levels of gulo compared to males. This may suggest an increased susceptibility to the effects of vitamin C supplementation for female mice. Overall, these data highlight vitamin C as a safe and well-tolerated potential therapeutic adjunct in a penetrant Mendelian disorder with high mortality. The mechanism for the sexual dimorphism noted in these experiments may relate to sex-limited endogenous production of vitamin C and perhaps some degree of crosstalk with pubertal androgens. These and other issues are currently being explored in gulo-null VEDS mice, and those results remain pending. We would like to thank the societies and foundations who've supported this research, and I'm happy to take any questions.
Thank you, Becky. Our last talk is by Zach Berger, and it's assessing the effects of aerobic and isometric exercise in vascular Ehlers-Danlos syndrome. Zach.
Thank you. Can you see my screen?
Yes.
Perfect. So like Dr. Dietz just said, I will be discussing the effects of aerobic and isometric exercise in a mouse model of vascular Ehlers-Danlos syndrome. So these are our disclosures. The current treatment strategies for patients with vascular Ehlers-Danlos syndrome, or VEDS, include blood pressure lowering drugs such as beta blockers and angiotensin receptor blockers. High-dose vitamin C is used to optimize the quantity and quality of the collagen matrix. There is also a general recommendation to avoid activities with a high degree of hemodynamic stress, such as vigorous isometric exercise, aerobic exercise to the point of exhaustion, and contact sports. The types of exercise and intensity of exercise permitted is an extremely common patient question. According to the American Heart Association, in the absence of an aneurysm or dissection of the aorta, or other major branch vessel, VEDS patients may participate in Class 1A sports such as bowling or golf. The general clinical recommendation is that all VEDS patients should avoid sports with a collision potential, perform low to moderate aerobic exercise, and any weight training should be done with very low weight and high repetitions. These recommendations are largely based on theory, highlighting the need for dedicated research to better inform people with VEDS. The effects of aerobic exercise have been studied in mouse models of other connective tissue disorders. A study in a Marfan mouse model explored the effects of voluntary exercise, which involved giving mice continuous access to a running wheel, as well as forced exercise, which utilizes a treadmill system. This study found that both exercise groups demonstrated attenuation of aortic root dilation and decreased elastic fiber fragmentation. This prompted us to explore the effects of aerobic exercise in a mouse model of VEDS. To explore this question, we utilized forced aerobic exercise using a treadmill system. As you can see here in the video, the mice are capable of exerting themselves to keep up at the maximum intensity. Acclimation to the treadmill was established with progressively longer duration and higher intensity over the course of 1 week, followed by a 4-week period of exercise 5 days per week, consisting of 30 minutes per day total, with 20 minutes at a top speed of 15 meters per minute. Wild-type mice were capable of completing the entire exercise program, while VEDS mice struggled to keep up for the first 3 weeks, as demonstrated by decreased exercise time with wide variability. The performance of VEDS mice was equivalent to their wild-type counterparts for the last 2 weeks of the trial. As you can see, we observed no significant difference in survival between VEDS mice that were forced to perform aerobic exercise, shown by the dashed green line, and VEDS mice that did not exercise, shown by the red line. This observation remained true even after the analysis was stratified by sex. While quantification of collagen content using picroserous red staining suggests a decrease in VEDS mice that performed aerobic exercise, this effect was modest and of questionable biologic and medical significance. We observed no differences in elastic fiber breaks or aortic wall width in association with aerobic exercise. We next assessed the impact of isometric exercise in VEDS mice. To model isometric exercise, we utilized inverted suspension, which required mice to maintain a grip supporting their body weight for an extended period. This had previously been validated as a form of isometric exercise by published animal physiology studies. Acclimation to this model was established over the course of 1 week, followed by a 4-week period of exercise 5 days per week with a duration of 10 minutes. Both wild-type and VEDS mice were capable of performing the entire exercise program from the onset of the study. As you can see, we observed no significant difference in survival between VEDS mice that were forced to perform isometric exercise, shown by the dashed green line, and VEDS mice that did not exercise, shown by the red line. When we stratify by sex, there is again no significant difference observed in male VEDS mice that performed isometric exercise, with perhaps a trend for early death. In striking contrast, female VEDS mice who performed isometric exercise demonstrated remarkable protection from aortic dissection, compared to female VEDS mice who did not exercise, with 100% survival in the exercise group, but only 50% survival in controls over the 40-day period of observation. In review, there are differential responses to different types of exercise in both sexes, with males showing a strong trend for worsened outcomes with imposed isometric exercise, compared with response to aerobic exercise. The exact opposite was seen in female mice with a survival benefit uniquely observed after isometric exercise. The sexual dimorphism seen in VEDS mice in response to different types of exercise is striking, but not yet mechanistically explained. We posit a potential role for androgens or other hormones that can be induced during exercise. Sex-specific differences in the production, processing, and response to androgens during exercise and chronic adaptation have been observed in both rodents and humans. Multiple hypotheses are currently being tested using pharmacologic provocations, including androgen and estrogen antagonists during and after exercise. Finally, I would like to thank my fellow lab members and acknowledge specific funding from the Ehlers-Danlos Society, as well as our other funders, and I would be happy to take any questions.
Okay, I'd like to thank everybody right now for this first session. Let's take a 5-minute break or come back at 5 to 12 and we'll start the next session. Okay?
Thanks, Joe.
Thanks.